Hello guys, today we are going to discuss about basic terminologies that are used in genetics. Okay, before starting the topic, we have to get to know what is genetics. Genetics is a branch of biology which deals with the principles of heredity and variation. Okay, now we'll discuss this. What is this heredity? Heredity generally means transmission of character from one generation to the next generation. Suppose he is the father and he is the son. Transmission of characters from one generation to another generation. That means transmission of character from father to the son. For example, if father is tall and son is also tall, this is called as heredity. If father is having black hair color, son is also having black hair color. This is heredity. If father is having brown eyes or black eyes and son is also having the same color of eye, then it is called as heredity. And variation. Variation generally means difference among the progeny of the same parent. Suppose this is the father and he is having two sons. The two brothers of the same parent won't be same, right? Like the facial shape or like anything, hair color or anything that may change. This change between the progeny uh, the change between the progeny is said to be the variation. This is called variation. We can simply say it as difference among the progeny of same parent. Okay, this is said to be variation. Now we will come to the allele. Before coming to the allele, we have to get to know uh, what is a gene. Gene means in any part of the chrome in our DNA. Sorry. In our DNA, some part of the nucleoside, like nucleotide sequence, will govern one character. This also govern like another character, right? How much this is governing the character? For example, this governs the height of the plant or color of flower. This governs some other character. This type of governance by this type of nucleotide sequence, this part is called gene. This gene generally governs one character. Now allele. Allele is simply alternative form of a gene. For example, gene is there Gene generally gives one character. For example, let's take height. Height of the plant. Height of the plant is of two types, right? One is tall, one is dwarf. Genes will also have two types of alleles. Then how many forms of this height are there, character is there, same that many forms of the genes will be there. Alleles, two types of alleles will be there, right? One is tall, capital T, one other is dwarf, small t. These are alleles. So we will say allele as alternative form of a gene. Okay. And locus, place on chromosome where gene is present. For example, this is the chromosome. The place where the gene is present in that chromosome is said to be the locus. And homozygous and heterozygous. As the name says it all, homo means same, hetero means different. Okay. Example, height of a plant. Height is of two types, right? Tall, dwarf, right? Tall is capital T, dwarf as small t. In any gene, if both the alleles are of same type, means this allele and this allele, these two alleles are of same type, right? Then it is called as homozygous, right? This is called homozygous dominant allele, and this is called homozygous recessive allele. Okay. And heterozygous, if different types of allele is present, then it is said to be heterozygous. Different types of allele means one dominant allele and one recessive allele. This is called as heterozygous. The term zygous is always used at the gene level. Okay. Now we will discuss about homogeneous and heterogeneous. Homo, again the name says it all. Homo means same. Hetero means different. Right? Homogeneous and heterogeneous. The genus generally says at population level. Population level. For example, this is a population. If all the plants in a population are same, then it is said to be homogeneous population. Here in homogeneous population, all the plants in the population should be same. See here, all the plants are capital A, small a. So we can say it as homogeneous population. If all the plants in that population is different, small a, capital A, capital A, capital A, small a, like this, if any population is there, then it is called as heterogeneous population. Here we have to remember one thing, where zygous will come, this is at gene level. Where genus will come, it is at population level, right? This thing you have to remember. Next is phenotype and genotype. As this also name suggests, phenotype means physical appearance of a plant. Genotype means it is based on the gene content. Yeah, for example, two plants are there, plant 1, plant 2, right? This is plant 1, this is plant 2, right? By seeing this, we can say this is tall, this is dwarf, right? By seeing this, we can say, so this is the physical appearance of the plant, so this is said to be the phenotype. Genotype, see, now genotype, this both the plants are there, right? This will come due to this small t allele present in that, and this may come from capital T or capital T small t. We can't say that by seeing. We can't see this is tall plant, we can say, but we can't say this which form of allele has been came to get this height of this plant. So it's based on the gene content that is present in this plant. So this is said to be the genotype, genetical, right? It's based on the gene content, right? Now we will come for selfing and crossing. Selfing and crossing, right? Selfing means let's draw one flower. This is ovary. These are anthers. For example, this is a flower. Stigma, anther, thalamus, ovary, right? If transfer of pollen from anther to stigma of same flower, then it is said to be selfing. If transfer of pollen grains from anther to stigma of different flower, this anther, if it comes and falls on different stigma of another flower, then it is said to be the cross pollination. This is another flower of different plant, then it is said to be the cross pollination. Now we will come for monohybrid cross. Mendel has done this monohybrid cross. If we are studying one character for our study, then it is said to be the monohybrid cross. For example, height of the plant. In F1 we will get capital T and small t. If like this anything height of the plant, here we have taken, if any one character if we have taken for study, then it is said to be the monohybrid cross, dihybrid cross. If we take two characters for our study, then it is said to be the dihybrid cross. For example, round and yellow. Round and yellow, it's generally represented as capital R, capital R, capital Y into small r, small y, right? If we are taking these two characters for study, round and yellow, then it is said to be the dihybrid cross. Okay, just few points to remember. In F1, in dihybrid cross, how the progeny will be, all are yellow and round, because the alleles which were capital R, capital Y will come from here, small r, small y will come from here. All the progeny will be capital R, small r, capital Y, small y. Here the dominating alleles are present. That's why all the F1 will be the yellow and round. F2 will be in the ratio 9:3:3:1. This is phenotypic ratio. This is phenotypic ratio, and 1:2:2:4:1. This one will be the genotypic ratio. For a dihybrid cross, the conclusion we will get from a dihybrid cross is law of independent assortment. This is the important point to be remembered. And try hybrid cross. If a study of inheritance between three pairs of contrasting characters means six characters, three pairs means six alleles of the character, then it is said to be the trihybrid cross. Example, if you are taking round, yellow, same, and plant head, example, tall. In F1, everything capital R, yellow capital Y, tall, capital T. Okay, smaller recessive form of alleles, small y, small t, right? If we are crossed with this one with this one, let it be P1 and let this all be P2. If we cross this, F1 we will get, right? This F1 will be round, yellow and tall. Why we are coming like this? See, let's take the alleles from P1. R, Y, T into this P2 alleles we will take now. Small r, small y, small t. We will get capital R, small r, capital Y, small y, capital T, small t. So the F1 will be round, yellow and tall. The F2 ratio is 27:9:9:3:3:3:1. This is the phenotypic ratio. And in F2, genotypic ratio is 27 types it will come. Okay, this is the thing. Now coming to the back cross, test cross and out cross. Okay, we'll discuss regarding these three. First is back cross. Back cross is generally we can say as crossing on F1 hybrid with either of its parents. For example, capital T is one parent, small t is one parent. We will get capital T, small t. This is the F1 generation. If we cross this F1 with any of its parents, then it is said to be the back cross, right? This is the simple. If we cross this F1 with any of these parents, then it is said to be the back cross. Same coming to the test cross. If these are the parents, F1 is capital T, small t. This is F1, right? If we cross this F1 with recessive parent, then it is said to be the test cross. We can say the test cross as crossing of the F1 hybrid with homozygous recessive parent. Then it is said to be the test cross. Outcross. Outcross means same two parents, again capital T small t, this is our F1. Right? If we cross this F1 with our dominant parent, then it is said to be the outcross. Here some important points to remember. All the progeny in the outcross are generally shows the dominant characters. For example, let's take here capital T, capital T, small t. This we have written here. This both capital T's we have written here. See, capital T, capital T, capital T, capital T, capital T, small t, capital T, small t. All are showing the dominant characters only. All the plants in this generation will be tall. So all the progeny of the outcross generally shows dominant characters. In test cross, 50% of the progeny shows dominant and remaining 50% shows recessive characters. This is generally in monohybrid cross, I'm saying. Just remember this point. And another point regarding this test cross and back cross, you have to remember this test cross and back cross. All the test crosses are back cross, but all the back crosses are not test cross, because in back cross, in back cross we can cross it with any of this parent, right? Any of this parent, dominant or recessive one. But in test cross we have to cross this with recessive one only. So we can say test cross is one type of back cross, but back cross is not test cross. This is the point you have to remember here. And the next thing is reciprocal cross. Reciprocal cross generally means changing of parents, right? If, for example, A and B are two parents, A is male generally means pollen we will take from here, B is female and we will take egg from here, right? And we will get the F1. And same, A and B parents, here we have taken pollen, right? Now we will take egg from here and consider this as female parent, and this we will take as male parent, and we will take pollen from here and we will get the F1. Just this changing the parents, like from male parent, A as male parent and A as female parent, just this changing of parents is generally we can say it as reciprocal cross. And again, only cytoplasmic characters, if it is anything is present, cytoplasmic characters means cytoplasmic genes, if it is covered, then only the F1, the change in the F1 will come, or else there won't be any change in the F1. Same thing that's why we will prefer for like half diallele compared to full diallele. Half diallele and full diallele, or later we can discuss this half and full diallele. Based on this simple concept only we will do this, the half and full diallele for the crops which cytoplasmic characters are not governing. Yeah, that's it. These are the reciprocal cross. And contrasting character. What generally contrasting characters means? we have to easily differentiate between these two characters. If we differentiate between two characters, then those characters are said to be the contrasting characters. Example, two plants are there, and see, this is plant 1, this is plant 2. We can easily say the height of plant 1 is more compared to the plant 2, right? This is tall, this is dwarf. Then this type of characters we can say it as contrasting characters. Mendel has taken 7 contrasting characters in his experiment. They are plant height, P shape, P color, pod shape, pod color, flower color, flower position. These are the 7 contrasting characters taken by Mendel in his experiments. That's it for today. Tomorrow again, next day we will continue with the another video. If you guys have any doubt, just comment down below. I will reply all those. Thank you.